Welcome friends. Today I am going to discuss the topic entitled Lipids, Fatty Acids, Triacylglycerols, Glycerophospholipids and Sphingolipids. The main objectives are to understand, to study the structure of lipids, to describe the classification of lipids, to describe the fatty acid structure and classification, to enlist the function of lipids, to study the structure and function of triglycerides and phosphoglycerides, to study the composition of sphingolipids. Lipids. The lipids are a heterogeneous group of compounds including fats, oils, steroids, waxes and related compounds, which are related more by their physical than by their chemical properties. Lipids are a class of compounds distinguished by their insolubility in water and solubility in non-polar solvents. Lipids are important in biological systems because they form the cell membrane, a mechanical barrier that divides a cell from the external environment. Lipids also provide energy for life, and several essential vitamins are lipids. Lipids can be divided in two major classes: saponifiable lipids and non-saponifiable lipids. A saponifiable lipid contains one or more ester groups, allowing it to undergo hydrolysis in the presence of an acid, base or enzyme. These include triglycerides, phospholipids, glycolipids, sphingolipids and the waxes. These lipids are known as simple lipids. A non-saponifiable lipid cannot be broken up into smaller molecules by hydrolysis, which include cholesterol and prostaglandins. Within these two major classes of lipids, there are several specific types of lipids important to life, including fatty acids, triglycerides, glycerophospholipids, sphingolipids and sterols. Each of these categories can be further broken down. Non-polar lipids such as triglycerides are used for energy storage and fuel. Polar lipids, which can form a barrier with an external water environment, are used in membranes. Polar lipids include glycerophospholipids and sphingolipids. Fatty acids are important components of all of these lipids. Biological role of lipids. Lipids have a common property of being relatively insoluble in water and soluble in nonpolar solvents such as ether and chloroform. They are important dietary constituents not only because of their high energy value, but also because of the fat-soluble vitamins and the essential fatty acids contained in the fat of natural foods. Fat is stored in adipose tissue, where it also serves as a thermal insulator in the subcutaneous tissue and around certain organs. Non-polar lipids act as electric insulators, allowing rapid propagation of depolarization waves along myelinated nerves. Combination of lipids and proteins, that is lipoproteins, are important cellular constituents occurring both in the cell membrane and in the mitochondria, and serving also as the means of transporting lipids in the blood. Knowledge of lipid biochemistry is necessary in understanding many important biomedical areas. Example: obesity, diabetes mellitus, atherosclerosis, and the role of various polyunsaturated fatty acids in nutrition and health. Now we will study classification of lipids. Lipids are classified into simple lipids, complex lipids, and precursor derived lipids. First, simple lipids. Simple lipids are esters of fatty acids with various alcohols. These include fats and waxes. Fats are esters of fatty acids with glycerol, while as waxes are esters of fatty acids with higher molecular weight monohydric alcohols. Now coming to complex lipids, they are esters of fatty acids containing groups in addition to an alcohol and a fatty acid. Complex lipids include phospholipids, that is, lipids containing in addition to fatty acids and an alcohol, a phosphoric acid residue. They frequently have nitrogen containing bases and other substituents. Example, in glycerophospholipids, the alcohol is glycerol, and in sphingolipids, the alcohol is sphingosine. Another complex lipid class is glycolipids. These are also known as glycosphingolipids. Lipids containing a fatty acid, sphingosine, and carbohydrate. The other complex lipids include sulfolipids and aminolipids. Lipoproteins may also be placed in this category. After complex lipids are precursor and derived lipids. These include fatty acids, glycerol, steroids, other alcohols, fatty aldehydes and ketone bodies, hydrocarbons, lipid-soluble vitamins, and hormones. Because they are uncharged, acyl glycerols, cholesterol, and cholesterol esterases are termed neutral lipids. Now we will move towards the second part of the lecture, that is fatty acids. A fatty acid is a molecule characterized by the presence of a carboxyl group attached to a long hydrocarbon chain. Therefore, these are molecules with a formula RCOOH, where R is a hydrocarbon chain. Fatty acids can be said to be carboxylic acids and come in two major categories. Saturated fatty acids do not have any double bonds. A fatty acid is saturated when every carbon atom in the hydrocarbon chain is bonded to as many hydrogen atoms as possible. Saturated fatty acids are solids at room temperature. Animal fats are a source of saturated fatty acids. In addition, fatty acids pack easily and form rigid structures. Example, fatty acids are found in membranes. Unsaturated fatty acids can have one or more double bonds along its hydrocarbon chain. A fatty acid with one double bond is called monounsaturated. If it contains two or more double bonds, we say that the fatty acid is polyunsaturated. The melting point of a fatty acid is influenced by the number of double bonds that the molecule contains and by the length of the hydrocarbon tail. The more double bonds it contains, the lower the melting point. As the length of the tail increases, the melting point increases. Plants are the source of unsaturated fatty acids. Classification of fatty acids. Naturally occurring fatty acids have cis bond. Trans fatty acids are created artificially using a process called hydrogenation. A trans fatty acid has a trans configuration rather than cis configuration at each double bond. This causes the molecule to straighten. These two stereoisomers can be distinguished in the following way. In a cis stereoisomer, two similar groups attached to the carbon double bond are found on the same side, while as in a trans stereoisomer, two similar groups attached to the carbon double bond are found on opposite sides. What are essential and non-essential fatty acids? If a fatty acid can only be obtained from the diet for humans, then the fatty acid is an essential fatty acid. Two fatty acids cannot be synthesized in human body and are therefore essential. These are linoleic and linolenic fatty acids, which are both unsaturated. Non-essential fatty acids can be made by the human body and so do not need to be obtained from diet alone. These are made from carbohydrates and proteins or from other fatty acids. Fatty acids are an important source of energy, while carbohydrates or proteins only provide 4 kilocalories per gram of energy. Fatty acids provide more than twice the energy per unit weight of 9 kilocalories per gram. This is one reason why a high-fat diet can lead to obesity. Fatty acids are aliphatic, carboxylic acids. Fatty acids occur mainly as esters in natural fats and oils, but do occur in the unesterified forms as free fatty acids, a transport form found in the plasma. Fatty acids that occur in natural fats are usually straight-chain derivatives containing an even number of carbon atoms. The chain may be saturated, containing no double bonds, or unsaturated, containing one or more double bonds. Now we will take on triacylglycerols, also known as triglycerides, are the simplest lipids formed by fatty acids. It is made up of 3 fatty acids ester linked to a single glycerol. Most triacylglycerols contain 2 or 3 different fatty acids. Those containing the same kind of fatty acids in all the 3 positions are called simple triacylglycerols and are named after the fatty acids present. However, majority of the naturally occurring triacylglycerides contain more than one type of fatty acid chains and hence called mixed triacylglycerides. These compounds are unambiguously named by specifying the name and position of each fatty acid. Triacylglycerols are nonpolar, hydrophobic, and insoluble in water. This is due to the ester-linked bonds between the polar hydroxyl of glycerol and the polar carboxylates of fatty acids. They also have densities lower than that of the water and do not mix with water, but form a separate phase and float on water. There are many different types of triglycerides, with the main division between saturated and unsaturated types. Triacylglycerols are vegetable oils, dairy products, and animal fat. Triacylglycerols are stored as fat droplets in large amounts in vertebrate fat cells and in plants as oils in the seeds. In most eukaryotic cells, triacylglycerols form a separate phase like oily droplet in the cytosol, serving as a deposit of metabolic fuel. Vertebrate animals have specialized cells called adipocytes that store large amount of triacylglycerols as fat droplets, which nearly fill the cell. Triacylglycerols is a better stored energy source than polysaccharides because oxidation of triacylglycerols produce more than twice as much energy that the oxidation of carbohydrates produce. In addition, due to triacylglycerols characteristics of being hydrophobic, it does not require hydration. Thus, it saves the organism the energy required to carry the extra water. Also, triacylglycerol located under the skin can function as insulation, in addition to being as energy source. Now we will try to understand another component of today's lecture, that is glycerophospholipids. Phospholipids in which the phosphate group and fatty acids are esterified to a glycerol are named phosphoglycerides, glycerophospholipids or phosphatides. These are the most common class of complex lipids and contain phosphoric acid residue, that is a phosphate group, and 2 fatty acid esterified to glycerol. Attached to the phosphate group is an amino alcohol, sometimes referred to as the nitrogenous base, which may be either serine, choline, or ethynylamine, or sometimes the monomethyl or dimethyl derivatives of ethanoline. Alternatively, a polyhydroxy compound, which is either glycerol, myoinositol, or one of their derivatives, is attached instead. Phosphoglycerides are derivatives of glycerophosphoric acid, which is also called phosphatidic acid. The different phosphoglycerides are often named by placing the constituents attached to their phosphate group after phosphatidyl. Example, phosphatidylcholine. There are many phosphoglycerides because of the possible variation in the fatty acid chains. The phosphoglycerides are good emulsifying agents because of the polar phosphate group and the presence of a group attached to it, which may also B charged, making one end of the molecule highly hydrophilic, while the rest of the molecule is composed of large hydrophobic groups. They are significant components of cell membranes and plasma lipoproteins, and also have very important physiological roles, including the prevention of the collapse of alveoli, which is made possible by the surfactant properties of mainly phosphatidylcholine. The final component of today's lecture is sphingolipids. Sphingolipids make the second largest class of membrane lipids. They have a polar head and two non-polar tails, but contain no glycerol. Sphingolipids are composed of, number 1, a molecule of long-chain amino alcohol, sphingosine, or one of its derivative. Number 2, a molecule of long-chain fatty acid. Number 3, a polar head alcohol. And number 4, sometimes phosphoric acid in diester linkage at the polar head group. A sphingosine molecule has OH, NH2 and OH group at C1, C2 and C3 position respectively, which are structurally homologous to the 3 OH groups of glycerol in glycerophospholipids. In the early days following their discovery, the biological functions of sphingolipids seemed as enigmatic as the Sphinx for which they were named. Now they are known to be involved in various recognition events at the cell surface. They are involved in receptor function of some pituitary glycoprotein hormones and toxicity of some bacterial toxins such as cholera toxin. They are also important components of specific receptor sites on the surface of cell membranes. For example, they are found in the specific sites on nerve endings to which neurotransmitter molecules become bound during the chemical transmission of an impulse from one nerve cell to the next. When a fatty acid is attached to C2 in amide linkage to the NH2, it is called as ceramide. That is, ceramides are N-acetyl fatty acid derivatives of sphingosine. Ceramides occur in small amounts in plants and animals, but are the parent compounds for more abundant sphingolipids such as sphingomyelins, cerebrosides, and gangliosides. Ceramide is structurally similar to diacylglycerol and is the fundamental structural unit common to all sphingolipids. There are 3 subclasses of sphingolipids, that is, sphingomyelins, neutral glycolipids, or glycophospholipids, and gangliosides. All of these are derivatives of ceramide but differ in their head groups. So we can summarize the present lecture by saying that the lipids are a heterogeneous group of compounds including fats, oils, steroids, waxes, and related compounds, which are distinguished by their insolubility in water and solubility in nonpolar solvents. With this, we conclude today's lecture: lipids, fatty acids, triacylglycerols, glycerophospholipids, and sphingolipids. Thank you very much.